Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Analytical Profile Index, API test. API 20E, 20NE, and 10S explained. Table of Content. Introduction to Analytical Profile Index Test. The principle of Analytical Profile Index Test. Common types of Analytical Profile Index systems. Biochemistry tests in Analytical Profile Index strips. Procedure. Example, Analytical Profile Index 20E. Interpretation and reading. The numerical profile, the coding system. Advantages and disadvantages. Mock calculation exercises. A. Analytical Profile Index 20E, Enterobacteriaceae example. B. Analytical Profile Index 20NE, non-enteric example. C. Analytical Profile Index 10S, rapid screening example. Conclusion. Introduction to Analytical Profile Index Test. The Analytical Profile Index is a standardized, miniaturized system used for the biochemical identification of bacteria and some yeasts. It consists of a series of small, dehydrated biochemical substrates arranged in a plastic strip, each designed to test a specific metabolic or enzymatic property of the microorganism. The API system was developed by Biomedical, a leading company in diagnostic microbiology, and is now a globally used tool for microbial identification. This system provides a rapid, reliable, and cost-effective alternative to traditional biochemical testing methods, which often require multiple media, larger volumes, and longer incubation times. Each API strip is tailored to a specific microbial group, e.g., Enterobacteriaceae, non-fermenters, gram-positive cocci, anaerobes, or yeasts, ensuring precise and targeted identification. The tests are interpreted based on color changes after incubation, and the resulting profile is converted into a numerical code known as an API. Profile number. This profile number is matched against an extensive database to accurately determine the genus and species of the microorganism. API systems are commonly used in clinical diagnostics, food microbiology, water testing, pharmaceutical quality control, and research laboratories. They require only a small inoculum, making them suitable when sample quantity is limited. The entire process is user-friendly, reproducible, and can deliver final identification results within 18 to 24 hours, depending on the strip type. Because of its simplicity and standardized nature, the API system is widely used for teaching microbiology and training laboratory personnel. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. The principle of Analytical Profile Index, API, test. The Analytical Profile Index, API, test works on the principle of evaluating multiple biochemical reactions performed simultaneously in a compact plastic strip. Each API strip contains a series of microcupules, tiny wells, filled with dehydrated biochemical substrates, each designed to test a specific metabolic or enzymatic activity of the organism. Rehydration. When a standardized bacterial suspension is inoculated into these wells, the dehydrated substrates become rehydrated, allowing biochemical reactions to begin. Metabolism. If the inoculated microorganism produces the enzymes required to utilize, degrade, or modify the substrates in a specific well, a metabolic reaction occurs. This can involve carbohydrate fermentation, amino acid breakdown, enzyme production, or utilization of specific compounds. Some tests may require anaerobic conditions, which are achieved by overlaying certain wells with mineral oil to block oxygen exposure. Visualization. As the reactions proceed during incubation, they produce visible color changes that indicate positive or negative results. In some wells, reagents must be added after incubation to reveal the final color reaction, e.g., Kovacs reagent for indole, ferric chloride for phenylalanine deaminase. The pattern of color changes forms a unique biochemical profile for the organism and is later converted into a 7-digit or 9-digit API code, depending on the strip type. This code is compared with an established database to determine the most probable identity of the microorganism. The principle relies on the fact that each bacterial species has a distinctive metabolic fingerprint, making the API system a reliable method for microbial identification. Common types of API systems. Different API systems are designed to target specific groups of microorganisms based on their biochemical characteristics and metabolic pathways. Each strip contains a defined set of tests optimized to identify organisms within a particular taxonomic or physiological group, ensuring more accurate identification. Common API systems used in microbiology laboratories include API 20E, Enterobacteriaceae, and other non-fastidious gram-negative rods. API 20NE, non-enteric gram-negative rods, such as Pseudomonas, Acinetobacter, Moraxella. API 10S, simplified version for Enterobacteriaceae, containing 10 essential biochemical tests. API Staph, designed for identification of Staphylococcus and Micrococcus species, focusing on enzymatic and carbohydrate metabolism. API Strep, used for Streptococcus, Enterococcus, and related genera through tests like enzyme activity and carbohydrate utilization. API 20A targets anaerobic bacteria, including Clostridium, Bacteroides, and others requiring oxygen-free conditions. API Candida, used for the identification of medically important yeasts such as Candida albicans, Candida tropicalis, and Cryptococcus species. The choice of API strip depends on the gram reaction, morphology, and preliminary biochemical traits observed during routine laboratory workup. In-depth. Look at key strips. A PI20E, one of the most widely used identification systems for Enterobacteriaceae. Tests focus mainly on carbohydrate fermentation, enzyme production, e.g., decarboxylases, deaminases, and substrate utilization, commonly used for identifying organisms such as E. coli, Klebsiella, Enterobacter, Salmonella, and Shigella. Produces results typically within 18 to 24 hours. API 20NE, specially designed for non-enteric, oxidase-positive or oxidase-negative, non-fermenting, gram-negative rods, like Pseudomonas aeruginosa and Acinetobacter baumannii. Includes assimilation tests that determine whether a microbe can use a compound as its sole carbon source, a key feature separating non-fermenters from enteric bacteria. Incubation may extend up to 48 hours due to slower metabolic reactions. A PI10S, a compact version of API 20E consisting of only 10 essential biochemical tests, often used for rapid screening, teaching, or settings where a full biochemical panel is unnecessary. useful for basic identification of Enterobacteriaceae when time, workload, or resources are limited. Download our microbiology study app made for researchers like you. Biochemistry tests in API strips. A. Basic tests. ONPG, orthonitrophenyl beta, D. Galactopyranoside. Substrate similar to lactose, does not require permease to enter bacterial cell. Reaction: ONPG, galactose plus orthonitrophenol, yellow to pale yellow. Indicator: B. galactosidase activity. Result: positive equals yellow, negative equals colorless. ADH, arginine dihydrolase. Reaction: arginine, citrulline plus N, H3, ornithine plus carbamoyl phosphate, ATP plus CO2 plus NH3. Indicator: phenol red, orange within first 24 hours equals positive. Enzyme: catabolic arginine, carbamoyl transferase, carbamoyl kinase. pH: range 6.8 to 8.4. Result: positive equals orange, negative equals no color change. LDC, lysine decarboxylase. Reaction: lysine, cadaverine plus C, O2, pH increase. Indicator: phenol red. Result: positive equals color change, usually purple, negative equals no change. ODC, ornithine decarboxylase. Reaction: ornithine, putrescine plus carbon dioxide, pH increase. Indicator: phenol red. Result: positive equals color change, negative equals no change. CIT, citrate utilization test. Reaction: sodium citrate, oxaloacetic acid, pyruvate plus carbon dioxide plus ammonia, alkalinization. Indicator: bromothymol blue, yellow pH 6, green 6.9, blue 7.6. Enzyme: citrate lyase, OAA decarboxylase. Result: positive equals blue dot green, negative equals yellow. H2S production. Substrate: sodium thiosulfate. Reaction: H2S gas plus Fe, 2 plus arrow. Ferrous sulfide. Black precipitate. Result: positive equals black precipitate, negative equals no color. URE, urease test. Reaction: urea arrow 2, NH3 plus CO2. Works in both aerobic and anaerobic conditions. Prefers anaerobic. Indicator: phenol red. Result: positive equals pink, negative equals no change. TDA, tryptophan deaminase. Reaction: tryptophan arrow indole pyruvic acid plus NH3 arrow. Ferric hydrazone with FeCl 3 plus HCl. Note: FeCl 3 plus HCl prevents false positives. Result: positive equals reddish-brown precipitate, negative equals no color. IND, indole test. Reaction: tryptophan arrow indole plus pyruvic acid plus NH3. Reagent: Kovacs reagent. Result: positive equals red/pink ring, negative equals no color. VP, Voges-Proskauer test. Reaction: sodium pyruvate arrow 2, 3 butynedyl arrow acetoin. Reagents: Barrett's A, alpha naphthol, plus B, potassium hydroxide. Result: positive equals red color within 10 minutes, negative equals no change. GEL, gelatin liquefaction test. Reaction: gelatin arrow, amino acids, gelatinase activity. Result: positive equals liquefied gelatin, negative equals solid. Sugar fermentation/oxidation tests. Fermentation: Enterobacteriaceae, Aeromonas, Vibrio. Reactions start at the anaerobic bottom arrow, red from bottom to top. Yellow at bottom equals weak, delayed positive. Oxidation other gram-negatives. Reactions start at the aerobic top, rid top to bottom. Yellow top plus blue bottom equals oxidative utilization, positive only for non-Enterobacteriaceae. B. Supplementary tests, needed only with multi-taxon codes. Oxidation test tube. Medium, 1% glucose plus nutrient agar plus bromothymol blue. Reaction detects oxidative carbohydrate utilization. Fermentation test tube. Medium. 1% glucose plus nutrient, agar plus bromothymol, blue plus sterile mineral oil. Reaction detects fermentative carbohydrate utilization. Note: if supplementary tests are used, delay reading of all results to 48 hours to allow complete development of reactions. Procedure. Example: API 20E. A. Preparation. Ensure a well-isolated, pure culture of the organism is obtained on a fresh agar plate, as mixed cultures will produce incorrect biochemical profiles. Select a single, well-defined colony using a sterile loop or needle to avoid contamination. Prepare a bacterial suspension by emulsifying the colony in sterile saline, 0.85% sodium chloride, or sterile distilled water, ensuring the mixture reaches the recommended turbidity, typically equivalent to a 0.5 Macfarlane standard. Proper turbidity is essential because an overly dense or dilute suspension can alter the intensity of biochemical reactions, affecting the accuracy of the final API code. Mix the suspension thoroughly to ensure an even distribution of bacterial cells before inoculation. B. Inoculation. Place the API 20E strip into the plastic incubation tray, ensuring that distilled water is added to the bottom of the tray to maintain a humid environment during incubation. This prevents the reactions from drying out. Use a sterile pipette to carefully dispense the prepared bacterial suspension into the tube portion of each microcupule. Avoid introducing bubbles, as they can interfere with proper rehydration. For tests requiring anaerobic conditions, apply a layer of sterile mineral oil over the cupule after filling the tube. These typically include ADH, LDC, ODC, H2S, and URE tests. The oil prevents oxygen exposure, allowing anaerobic reactions to proceed correctly. For tests requiring aerobic conditions, fill only the tube portion and leave the cupule exposed to the air. This allows oxygen-dependent biochemical reactions to occur naturally. Ensure all wells are properly filled and that no overflow or cross-contamination occurs between adjacent cupules. Gently close the incubation tray lid to maintain a stable microenvironment throughout the incubation period. C. Incubation. Incubate the inoculated API strip at 37 degrees Celsius, which is the optimal temperature for most Enterobacteriaceae and other common gram-negative rods tested with API. 20E. Maintain the strip inside the closed incubation tray to preserve humidity and prevent evaporation of the biochemical reactions. Ensure the tray is placed in an incubator with stable temperature control, as fluctuations may slow down or alter metabolic reactions. Standard incubation time ranges from 18 to 24 hours, after which most biochemical reactions will have developed sufficiently to be interpreted. Some reactions may appear earlier, but reading the results before the recommended time can lead to false or incomplete reactions. If certain tests require delayed reading, such as VP, follow the manufacturer's instructions for timing and reagent addition after incubation. Once incubation is complete, proceed immediately to add required reagents and interpret the results to prevent over-incubation, which may cause color fading or false positives. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Interpretation and reading. After completing the incubation period, each microcupule is examined for visible color changes that indicate whether the biochemical reaction is positive or negative. The color changes reflect specific metabolic activities of the bacterium, such as fermentation, enzyme production, decarboxylation, deamination, or compound utilization. Spontaneous reactions. Some tests develop their final color without the need for additional reagents. For example, the GLU, glucose fermentation test, turns yellow when acid is produced, indicating carbohydrate fermentation. Other spontaneous reactions include ONPG, ADH, LDC, ODC, CIT, H2S, and URE, each with characteristic color outcomes. Reagent-dependent reactions. Certain tests cannot be interpreted until specific reagents are added after incubation. These reagents interact with metabolic end products created by the bacteria to produce the final detectable color. TDA, tryptophan deaminase. Add ferric chloride, FeCl3, to the well. A positive reaction appears as a brown to reddish-brown color due to the formation of ferric complexes. IND, indole test. Add Kovacs reagent. A positive result produces a red ring at the surface, indicating degradation of tryptophan to indole. VP, Voges Proskauer test. Add Barrett's reagent A, alpha naphthol, followed by reagent B, KOH. A positive result develops a pink to red color, confirming acetoin production via the butyridyl fermentation pathway. Each color reaction should be interpreted within the recommended time frame, as delayed reading may cause colors to fade, intensify, or shift, leading to incorrect identification. After all wells are interpreted, the positive and negative reactions are recorded to generate the final numerical profile for organism identification. The numerical profile, the coding system. One of the defining advantages of the API system is its ability to convert numerous biochemical test results into a single, easy-to-interpret numerical code. This coding system transforms qualitative outcomes, positive or negative reactions, into quantitative values, creating a standardized identification method. The coding is based on the triplet system, in which the 20 biochemical tests are arranged into groups of 3 consecutive tests. Each test within a triplet is assigned a numerical value if the reaction is positive. The first test in the triplet has a value of 1. The second test has a value of 2. The third test has a value of 4. If a test is negative, it receives a value of 0. The sum of the values for each triplet generates a single digit, and these digits collectively form the 7-digit, or longer, API profile code. This conversion system ensures that each unique biochemical pattern corresponds to a specific numerical signature, making microbial identification simple and reproducible. Example calculation. Tests in one triplet, ONPG plus, ADH plus, LDC minus. Values assigned, ONPG plus, 1 ADH plus, 2 LDC minus, 0. Summation, 1 plus 2 plus 0 equals 3. This value becomes the first digit of the final API profile code. After all triplets are calculated, the resulting numerical code is either checked in the API codebook, compared with the reference index, or entered into the API Web software. API Web uses a large database of biochemical profiles to provide the most probable bacterial species, along with confidence percentages and alternative identifications, if applicable. This standardized system significantly reduces human error and enhances accuracy in microbial identification. Advantages and disadvantages. Advantages. Standardized. API strips are manufactured under strict quality control, ensuring consistent results across different laboratories, countries, and personnel. This standardization improves interlaboratory comparability and reduces variability seen in conventional biochemical tests. Fast identification. Most organisms can be identified within 18 to 24 hours, which is significantly quicker than traditional biochemical tube methods that may take several days. Rapid turnaround time is especially useful in clinical diagnostics, food safety testing, and quality control. Comprehensive testing. A single API strip evaluates multiple biochemical properties simultaneously, giving a broad metabolic profile of the organism. This allows the identification of organisms that require many tests, saving time and resources. Long shelf life. The dehydrated substrates in each cupule remain stable for extended periods without the need for refrigeration. This makes storage easy, reduces waste, and ensures strips are ready for use whenever needed. User-friendly and minimal equipment. Required. Only basic tools like saline, a pipette, and an incubator are needed, making it ideal for small labs, teaching labs, and resource-limited settings. The interpretation system, numerical coding, simplifies identification. Disadvantages. Cost. API strips are more expensive per test compared to conventional biochemical tubes or in-house media preparation. This may be a concern for high-volume laboratories or institutions with limited budgets. Requirement for pure culture. Mixed cultures cannot be tested, as even a small contamination can alter the biochemical profile and produce incorrect identification. Additional steps, subculturing, streaking, are needed to ensure purity. Database limitations. Identification is restricted to organisms that exist in the API database. Rare, newly discovered, or atypical strains may give low confidence scores or no match. Subjective interpretation. Some color changes can be faint, ambiguous, or variable, depending on incubation time and lighting conditions. Misreading colors may lead to incorrect numerical codes and false identifications. Certain organisms require longer or special conditions. Some bacteria, especially non-fermenters or slow-growing organisms, may require extended incubation times or additional confirmatory tests. Mock calculation exercises. These exercises illustrate how to calculate the final numerical profile for different API strips. AAPI 20E Enterobacteria CAE example. Scenario: identification of an Enterobacteria CAE organism. Profile: 7 digits, 6 triplets, plus 1 pair. Now let's calculate the numerical profile based on the triplet system used in API 20E. Triplet 1. ONPG is positive, so it gets a value of 1. ADH is negative, so it gets 0. LDC is positive, so it gets 4. Adding these gives the first digit, 5. Triplet, 2. ODC is positive, giving 1. CIT is negative, 0. H2S is also negative, 0. So the second digit is 1. Triplet, 3. URE is negative, 0. TDA is positive, 2. IND is positive, 4. The total is 6. Triplet 4. VP is negative, 0. GL is positive, 2. GLU is positive, 4. Together, these give 6. Triplet 5. Man is positive, 1. INO is negative, 0. SOR is positive, 4. Adding them gives 5. Triplet 6. RHA is negative, 0. SSC is positive, 2. MEL is positive, 4. The sixth digit is 6. Pair. 7. AMY is negative, 0. ARA is positive, 2. So the final digit is 2. Putting all digits together, the final API 2. 0E profile code is 5166562. According to the API identification database, this biochemical profile corresponds to Escherichia coli. BAPI 20NE non-enteric example. Scenario: identification of a Pseudomonas-like organism. Profile: 7 digits, 6 triplets plus 1 pair. Now let's calculate the numerical profile for the non-enteric organism using the API 20NE system. Triplet 1, NO3 is positive, so it gets a value of 1. TRP is negative, 0. GLU fermentation is also negative, 0. Adding these gives the first digit, 1. Triplet 2, ADH is positive, giving 1. URE is negative, 0. ESC is negative, 0. So the second digit is 1. Triplet 3, GEL is positive, giving 1. PNPG is negative, 0. GLU assimilation is positive, giving 4. The total for this triplet is 5. Triplet 4, ARA is negative, 0. MNE is negative, 0. MAN is also negative, 0. This results in the fourth digit, 0. Triplet 5, NAG is negative, 0. MAL is negative, 0. GNT is positive, giving 4. So the fifth digit is 4. Triplet 6, CAP is positive, 1. ADI is positive, 2. MLT is negative, 0. Adding these gives the sixth digit, 3. Pair 7, CIT is positive, giving 1. PAC is positive, giving 2. Together, these give the final digit, 3. Putting all digits together, the final API 20NE profile code is 1, 1, 5, 0, 4, 3, 3. According to the API identification database, this biochemical pattern is characteristic of Pseudomonas aeruginosa. CAPI 10S, rapid screening example. Scenario: quick identification of Enterobacteriaceae. Profile: 4 digits, 3 triplets plus 1 single. Now let's calculate the numerical profile for the rapid identification using the API 10S system. Triplet 1, ONPG is positive, giving a value of 1. GLU is positive, giving 2. ADH is negative, giving 0. Adding these gives the first digit, 3. Triplet 2, LDC is negative, 0. ODC is positive, giving 2. CIT is negative, 0. So the second digit becomes 2. Triplet 3, H2SE is negative, 0. URE is negative, 0. TDA is also negative, 0. The total for this triplet is the third digit, 0. Single test, IND is negative, giving 0. So the fourth and final digit is 0. Putting all digits together, the final API 10S profile code is 3200. According to the API identification database, this biochemical pattern is characteristic of Shigella sonnei. Conclusion. 1. What does API stand for? Analytical Profile Index. 2. What is the most common API strip? A PI20E, used for Enterobacteriaceae. 3. What is API 20NE used for? Identification of non-enteric gram-negative rods, e.g. Pseudomonas acinetobacter. 4. What is the main difference in 20NE tests? It includes assimilation tests, ability to use compounds as a carbon source, not just fermentation. 5. Why is mineral oil used in some wells? To create anaerobic conditions for reactions that are inhibited by oxygen. 6. How is a bacterium identified using API? By generating a numerical profile code based on positive/negative results and comparing it with the API database or API web software. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you.